I'm SARIGA JAYADEVAN, Assistant Professor, Department of Biotechnology, AJK College of Arts and Science. Now in this session, we are going to discuss about the topic biosignaling. It is a process by which two cells communicate in our system. Why it is called biosignaling? Because it is taking place in our body. That's why it is called biosignaling. Now in the session, we can discuss about the type of biosignaling processes and the direction and the duration and how the process is taking place. Mainly based on the type or based on the direction, we can classify the process into two types, in-out signaling and out-in signaling. What is meant by in-out signaling? In-out signaling is nothing but where the information is creating within the cell and it is communicated out to the cell. But in out-in signaling, where the confirmed information is originating outside the cell and which is communicating into the cell. So based on the direction, we classify them into in-out or out-in. Now let's discuss about the skeleton of a signaling system. We know that for every communication there should be a flow. Likewise, biosignaling also have a flow that starts with an information carrying molecule which is called a ligand. This ligand is received by a receptor and this based on the ligand receptor interaction, a series of molecules are get activated. Either or they may be get deactivated. And these molecules which get activated or deactivated are collectively called the effector molecule. After these effective molecule activation or sometimes maybe it is deactivation, whatever it is, it will result in a response. So this is the skeleton, that is a receptor, ligand, receptor, effector molecule and a response. Now we can discuss more about each term. First is ligand. What is a ligand? Ligand is nothing but it is an information carrying molecule. Which kind of molecule can be act as a ligand? Fine. Any molecule can be act as a ligand. It may be either a protein or a liquid or a carbohydrate or any molecule. Even a conjugated protein can also act as a ligand. What is meant by conjugated protein? Nothing but a protein which is associated with any other non-protein molecule like carbohydrate or lipid or whatever it is. So that molecule can also be like act as a ligand. Now, based on the chemical nature, we can classify ligand into two types. First type, ligand which can cross the plasma membrane and the ligand which cannot cross the plasma membrane. What is the difference? Actually, we know that plasma membrane is a lipid bilayer, right? Lipid bilayer means it is made up of lipids. So, those molecules which can cross this plasma membrane means those molecules which can be dissolved in this plasma membrane, right? Plasma membrane is lipid nature. So, those which can dissolve in lipid can cross the plasma membrane. Or we can say that these molecules or those molecules which can cross the plasma membrane is essentially of hydrophobic in nature. What is meant by hydrophobic? Nothing but they are water not soluble in water. Or they are lipid soluble molecule or water fearing molecule. Phobic means fearing or fear. Phobia means fear. So, water soluble means that those which can dissolve in water or phobia means that those which cannot dissolve in water. So here those molecules which cannot cross the plasma membrane means essentially they are lipid soluble or water phobic or hydrophobic molecule. So ligand can be classified as either water soluble, that means hydrophilic, or which cannot cross the plasma membrane, or water lovable or water soluble, or which can cross the plasma membrane. So this is the classification of ligand. Now come to the receptor. Likewise, the ligand, any molecule can act as a receptor. It may be either a protein or a lipid or a nucleic acid or a carbohydrate. Any molecule can act as a ligand. Mostly 90% of the receptors are of protein in nature. Now, these receptors can also be classified into three types based on the position where we can see these receptors. First is transmembrane receptor. Second is receptor which is present inside the cell, which is called as intracellular receptor, or the receptor which is nuclear in nature. That we'll discuss shortly. Okay, what is about the nuclear receptor? There is a big class of nuclear receptor and the family of nuclear receptor with the signaling mechanism and all that we will discuss shortly. But for time being, we can just give the name only that they are nuclear receptors. And first is the transmembrane receptor. What is meant by transmembrane receptor? Transmembrane itself means that it is traversing through the membrane or transporting through the membrane. That means that type of receptors has two essential parts. First is a ligand binding unit and second is a catalytic binding unit. Obviously, its ligand binding unit is present above the membrane so that those molecules which cannot cross the plasma membrane can bind with this receptor and cause the that interaction or cause the process, whatever it is. And the catalytic unit is actually responsible for elicitate the reaction. Now, the intracellular receptors, those molecules which can cross the plasma membrane can be bind with this intracellular receptor. They can bind with the transmembrane receptors too, but these kind of molecules normally prefer intracellular receptors. The next one is nuclear receptors. That name means nuclear receptor, but that doesn't mean that they are present inside the nucleus. They may be present either in the nucleus or in the cytoplasm. But then why they are called nuclear receptors? Because their action is always on the nucleus. That's why they are called nuclear receptors, because these receptors, the activation of these receptors may result in the gene expression. Genes are always present in the DNA. DNA is present in the nucleus. That's why they are called nuclear receptors. Now about the effector molecule. A series of molecules, I already told that there are series of molecules which are get activated when a receptor interacting with a ligand. These molecules are called effector molecule. Based on the chemical nature or the type of reaction they elucidate, these molecules can again be classified into 4 types. First, adapters or bifurcators or integrators or amplifiers. What is meant by an adapter? Adapter is nothing but some molecule only doing the adapting of 2 molecules. That is, they are just acting as a binding or binding molecule between 2 molecules. That is called adaptation molecule or adapter molecule. Next is the integrators. Integrators means nothing but they are integrating 1 or 2 pathways that we will discuss that kind of molecule in the later stage of the same session. They are integrators. Bifurcators means they are essentially receiving some signals and bifurcating the information to several other pathways. Actually, all the pathways that are taking place in our body or in our system is actually they are interconnected. There is not a single pathway. Lots of pathways are taking place together at the same time. So they are interconnected. For interconnecting such different pathways, we need certain molecules, and that molecules are called integrators and the bifurcators. And amplifiers. Amplifiers, because of amplifiers only, we are actually getting a result, we're getting a reaction or a response. Why? Because we know that only nanomoleculars or nanomoles of ligand is coming and interacting with the receptor. But we are getting a notable result, right? See, for example, if insulin is producing, when insulin is producing only when there is an increase in the glucose concentration in our blood. When the glucose concentration increase in our body, when after a meal or after taking our food, our glucose level in our bloodstream increases. Within increase of this glucose level, our body producing insulin. But the nanomolecules or nano concentration of insulin is producing from our body, but it is creating a notable result, right? It creating a big effect in our body. This is only because of the amplifier molecule present in our signaling system. Last one is response. What is meant by response? Response means the outcome. What is the outcome of this reaction? Say, for example, in the case of insulin production, insulin is produced from our body, and what is its result? It results in the decrease in the glucose level in our bloodstream, or it converting excess glucose to glycogen. That is the result. So that is called the response. Based on the type of response or based on the chemical nature of response, we can again classify response into 2 types. First is biochemical response, second is the gene level response. What is called a biochemical response? Biochemical response is nothing but here the response involves the conversion of an inactive molecule to active molecule. And sometimes our body already produces some proteins or some enzymes and kept that enzymes in an inactive form. When there is a need, then only our body converts that inactive molecule into active form, which is called a biochemical response. But for gene level response, where we need to produce an enzyme, because that enzyme, the gene is there and the DNA is there, but that protein is not there at that time. But when there is a need, on that time, the body need to produce the enzyme. That is called the gene level response. So this is a skeleton for a signaling system. Now let's have a more detail about this each and every time. First is ligand. I already told you that ligand we can classify it into those which can cross the plasma membrane or those which cannot cross the plasma membrane, like that. Apart from that, based on the distance they can travel, we can again classify these ligands into 3 types. First is endocrine signals or endocrine ligands, then paracrine ligands and autocrine ligands. What is called endocrine ligand? Endocrine ligand is nothing but a ligand which is secreted into the bloodstream, and through the bloodstream it reaches a target cell. If it is a target cell which is far away from the original cell or the cell from where it is producing, means such kind of ligands are called endocrine ligand. Say for example, our hormones are coming under endocrine ligands. Class of endocrine ligands. Next is the paracrine. Paracrine means the ligand which is producing from the target cell, which is acting upon the nearby cell, that is neighboring cell. That is called the paracrine ligands. Some autocrine ligands are there. Autocrine ligand is nothing but the ligand, the information molecule which is producing from the cell acting on the same cell. That is self-acting molecule. Such kind of ligands are called the autocrine ligand. The best example for autocrine ligands are neurotransmitters like GABA or acetylcholine. All these are actually autocrine ligands. So these are the classification of ligands based on the direction that they can travel. Now, based on the type of receptor or receptor mechanism, we can broadly classify the whole biosignaling process into 5 types. First is receptor enzyme type signaling, or receptor with no intrinsic enzymatic activity, G protein coupled receptor signaling system, or nuclear receptor signaling system. Actually, what is meant by that receptor enzyme type? The first one is receptor enzyme type. The best example for receptor enzyme type signaling is the insulin signaling system. In receptor enzyme type, here the receptor itself has some enzymatic activity. That is the essential of this receptor enzyme type, where the receptor possessing some enzymatic activity, and the receptor is the first molecule which catalyzes the whole reaction. The main classes coming under this receptor enzyme type is RTK receptors. What is meant by RTK receptor? RTK means receptor tyrosine kinase. What is meant by receptor tyrosine kinase? Nothing but the receptor which is showing a receptor tyrosine kinase activity. Tyrosine kinase activity means nothing but the receptor which is doing the kinase function on the tyrosine residue of the target cell. Those receptors which can kinase on the tyrosine residues of the target cells, means such kind of receptors are called receptor tyrosine kinase. Okay, RTK. These RTKs are normally monomer in nature, and under the influence of ligands only they get dimerized. And they can show 2 types of dimerization. First, ligand mediated dimerization. Second, receptor mediated dimerization. Ligand mediated dimerization means where the ligand is responsible for the dimerization of the receptor monomer. As I already told that these these receptors are actually monomers, and they need to be get dimerized before the activation or for the function of the functioning of the signaling system. So, in the first case, that is ligand-mediated dimerization, where the ligand is responsible for the dimerization of the receptor. Normally, in such cases, ligand is a dimer, and the ligand bind with one of the monomers of the receptors, and that monomer undergo a dimerization, or that monomer first undergo a conformational change. That conformational change creates a docking site in a monomer. To that monomer, another monomer unit come and bind and form a dimer. This is the first case, that is in the case of ligand mediated dimerization. But in the second case, where there is a receptor mediated dimerization, where ligands are actually monomers, each monomer bind with each ligand molecule, and these receptor undergo dimerization, undergo a conformational change, and this conformational change creates docking site in each monomer's unit of the receptors, and the receptor get dimerized. This is called the receptor mediated dimerization. All the RTK receptors except insulin receptor as a dimer, existing as dimer. Only that insulin receptor existing as a dimer. Okay. Now, how that insulin pathway takes place, or what is meant by this enzyme, receptor enzyme type signaling mechanism. As I already told that receptor enzyme type means the receptor itself has some enzymatic activity, right? Now, once RTK receptor itself has some enzymatic activity, means In such case, the receptor is the first molecule which catalyzing the preceding molecules. In the case of insulin reaction or insulin cascade, when insulin is producing only when there is an increase in the concentration of glucose in our bloodstream. When there is an increase in glucose in bloodstream, the essential thing that should happen is to convert that glucose into glycogen. But the insulin pathway has two functions. First, conversion of glucose to glycogen. Second one, it has got some mitogenic activity. What is meant by mitogenic activity? It is nothing but it has got some cell renewal activity or cell cycle activity. It can promote some cell cycle reactions. Okay, first is the insulin act as a mitogen. How insulin can act as a mitogen or how insulin can promote cell cycle, that we can discuss first. Later we can discuss about how insulin can reduce the level of glucose in our bloodstream. Okay, first, once insulin come to bind with the insulin receptor. Insulin receptor is actually an RTK receptor. It is already existing as a dimer. Here we can discuss first about the structure of the insulin receptor. Then we can come to the signaling mechanism. What is the structure of an insulin receptor? Insulin receptor is a dimer. It is actually a transmembrane receptor. Transmembrane receptor means it has got three main important parts. First is a ligand binding domain, then a catalytic domain and a traversing domain. The traversing domain is actually responsible for that conducting the information from the ligand binding site to the catalytic site. Okay, its catalytic domain is found above the plasma membrane, its ligand binding domain is found above the plasma membrane, while its catalytic binding domain is found within the cell. Okay, once insulin comes and binds with the insulin receptor, actually insulin is coming as monomer. Here the insulin the insulin receptor is already a dimer, there is no ligand mediated dimerization or a receptor mediated dimerization. It's already existing as a dimer. Okay, fine. Once insulin comes and bind with the insulin receptor undergoes a dimerization, or it undergoes actually a conformation change. This conformation changes causes formation of activation of transverse, that is the RTK domain in the receptor. It is actually basically a receptor tyrosine kinase. So once the ligand bind with the receptor, here the ligand is insulin. Once the ligand bind with the receptor's RTK domain get on. What happened is, this RTK domain causes the trans-autophosphorylation of its domains. What is meant by trans-autophosphorylation? Let us assume a ligand with the two domains. Okay, one domain causes the phosphorylation of the second domain, while at the same time the second domain causing the dimerization or the phosphorylation of the first domain. This is called a trans-autophosphorylation. That is, first monomer causing or doing the phosphorylation of the second monomer, while the second monomer phosphorylating the first monomer. This is called a trans-autophosphorylation, and this is a particular reaction of RTK receptors. So this is very, very important, this autophosphorylation. Once insulin bind with the insulin receptor, the receptor's RTK activity become on and it causes the autophosphorylation of the receptor. This autophosphorylation creates some conformational change in the receptor. Why it undergoes a conformational change? See, here we are adding 6 phosphate group to each monomer, right? First monomer adding 3 phosphate group to the second monomer. At the same time, second monomer adding 3 phosphate group to the first monomer. Altogether, about 6 phosphate group is now adding. Each phosphate group has got a negative charge. That means now we are adding 6 negative charge to the receptor. This 6 negative charges causes the conformational change within the protein molecule. Protein molecule means here is the receptor. Now, in the receptor, one more additional site is formed, which is called a docking site. Docking site means nothing but the site which is for binding with another molecule. Here, this docking site is formed within the receptor, and this docking site is very particular for those molecules which is having PTB domain. Here in the cytosol, there are lots of protein molecules are there, but normally that molecules are not at all active. Why? Because there is no reaction is taking place, since that molecules are present within different lipid wrap. What is lipid wrap? That we will discuss shortly. Okay. Let it be there. Lipid raft, they are present in the different lipid raft, either in the same raft or in a different raft, whatever it is. They are all present in our cytosol. Normally, there is no interaction between this cytosolic protein molecule and a receptor. Why? Because there is no docking site or binding site in our receptor. But once the receptor creates a docking site, that docking site is very specific. That docking site is specific for those molecules with the PTB domain, right? Here in cytosol, there are lots of protein with the PTB domain. One of which is ISR. ISR means insulin substrate receptor 1. This now particularly bind with this docking site and create a full molecule. That is, now the receptor interacting with IRS1 and forms a full molecule. Then what will happen? Now the receptor itself having some RTK activity, right? Receptor kinase activity. What this receptor does, it will phosphorylate tyrosine residues present within this IRS1 molecule. That's why this signaling is called a receptor enzyme type. Now the receptor itself has some enzymatic activity. That's why that receptor is phosphorylating this IRS1. Now that IRS1 get phosphorylated, as we know that phosphate group means adding one more negative charge. Once it got negative charge, it cannot no longer interact with the receptor. Now, what happened is, now the IRS1 detached from the receptor and it enter into the cytosol. Now itself it is in the cytosol, but it is interacting with the docking site. After getting a negative charge, it reaches the cytosol where it interact with another molecule called GRB2. GRB2 means growth factor receptor binding protein 2. GRB2 is a protein molecule which is very specific for proline-rich domain region, where what happened is, once IRS1 phosphorylate this GRB2 goes and phosphorylate another molecule called SOS. SOS means son of 7S, another protein molecule called the son of 7S. Actually, this direct substrate of IRS1 is SOS, not GRB2. Here GRB2 is acting as an adapter. I already told that what is adapter. Here the exact function of GRB2 is adapting or to bringing these two molecules together, or IRS1 need to phosphorylate this SOS, and that is IRS1 cannot directly bind to SOS. So what it done is, through GRB2 it is binding with SOS and phosphorylating SOS1. Okay, now SOS has got two regions. First is a proline-rich region. Through this proline-rich region, actually GRB2 interacting with SOS. And once GRB2 interact with the SOS1 and this interaction causing the phosphorylation of SOS1 by IRS1, and that phosphorylation created docking site for RAS molecule. RAS is actually a proto-oncogene which now bind with this SOS1. Once RAS bind with SOS, RAS get activated via the same phosphorylation. Now the RAS, what it done is, it convert its GDP molecule in the normal inactive form. This RAS is a monomeric G protein. What is a G protein? G protein is nothing but a protein which is associated with a GDP or GTP or guanosine nucleotide is essentially called a G protein. RAS is a monomeric G protein. Basically, G proteins are two types. First is heterotrimeric G protein and the second one is monomeric G protein. Heterotrimeric means nothing but a G protein which has got three different monomeric unit, means such type of G proteins are called heterotrimeric G protein. While monomeric G protein means it has got only one domain. That essential domain is called alpha domain. Okay. Anyway, if a G protein which has got an alpha domain means such kind of G protein is called a monomeric G protein. This monomeric G protein is always associated with a guanosine nucleotide. Guanosine nucleotide means normally GTP, GDP or GMP. In normal cases, a G protein is always associated with a GDP in its inactive form. But once it gets activated, its GDP is replaced by GTP. But don't get confused that it is a phosphorylation reaction. It is not at all a phosphorylation reaction. It is just a replacing reaction, where the G protein replacing its GDP with the GTP. But now what happened is this RAS become activated. Once RAS become activated, it goes and interact with a series of molecules, and that molecules are actually called mitogen activated protein kinase molecule that we'll discuss. Okay. Now, what this RAS will do is, it will goes and interact with a molecule called RAF. Now, the RAF get phosphorylated and RAF get activated. Once RAF is activated, RAF goes and activate another molecule called RAC. Now, RAC goes and activate another molecule called ERK. Once ERK is get activated, that is the turning point in this reaction. Once ERK get activated, the function of ERK is to enter into the nucleus. Till before the activation, it cannot cross the plasma membrane. Why? Because it is not having any negative charge. Now it has got a negative charge. So what it done is, it enter inside the nucleus where it interact with a protein called ELK. And it phosphorylate this ELK. Once ELK is phosphorylated, ELK dimerize with another nuclear protein called SRF, serum response factor. Now, this ELK SRF form a dimer. This dimer is very important. This dimer acts as a mitogen activator protein, or we can say simply as a transcriptional activator of proteins involved in cellular function or involved in cell cycle, like JUN, FOS, or JUN, JUN like proteins. And these genes causes the cell cycle. That's why we can say that insulin is acting as a mitogen, or insulin has got a mitogen activity. Here, the production of insulin causes the formation of proteins which are essential for cell cycle. Okay, now come to the ELK. ELK is the dimer, or ELK is the protein which causes SRF dimerization and act as the transcriptional activator. Now that is the protein actually causing this transcription, right? Transcription of what? Transcription of this mitogenic protein like JUN, FOS, something. Okay, so this ELK is called a mitogen activator protein. Now this ELK is phosphorylated or activated by ERK, right? So, ERK can be called as mitogen activated protein kinase. Why it is called protein kinase? Because here ELK causing the phosphorylation of the proteins needed for cell cycle. So, they are called mitogen activated protein. How these proteins are activated? These proteins are activated by ERK. So, ERK is the kinase which causes the activation of this ELK. That's why ERK is called a mitogen activator protein kinase. Actually, that ERK is phosphorylated or activated by this RAF. So, RAF is called a mitogen activator protein kinase. The kinase, which kinases, the kinase responsible for the kinase reaction. That is called the kinase family of proteins. So, these about the mitogenic activity of insulin. But we know that the essential function of insulin is the conversion of glucose to glycogen. That is the primary function of glucose or insulin. How it is, how insulin performing that function. Now, let's the first point. First, insulin reacting with the insulin receptor and causing dimerization and something that we already discussed. During that time, we told that IRS1 is coming and interacting with the receptor and causing the undergo a dimerization and undergo a phosphorylation and getting activated. This IRS1, the first function of IRS1 or primary function of IRS1 is to goes and interact with the GRB2 and causes the mitogenic function. The second function is this IRS1 now goes and interact with another cytosolic protein called PI3K, phosphatidylinositol 3 kinase protein, which is shortly known as PI3K, phosphatidylinositol 3 kinase. The function, which is also a kinase, right? Name itself is implying that it is a kinase. So its function is it goes and interact or it goes and phosphorylate another protein which is a plasma membrane protein called PIP2. PIP2 is nothing but phosphatidylinositol 4,5 biphosphate or bisphosphate. Okay. So, this PI3K goes and phosphorylate a cytosolic protein called PIP2 and convert this PIP2 to PIP3. PIP3 is nothing but phosphatidyl inositol 3,4,5 triphosphate. Initially it is 4,5 diphosphate. This PI3K adding one more phosphate group and converting it to 3,4,5 triphosphate. Now this PIP3 interact with another cytosolic protein called PKB, phosphatidyl inositol triphosphate, interacting with the protein kinase B and activating this protein kinase B. There are two functions for protein kinase B. Once it is activated, it causes the opening of LUT4 channels and entering of glucose into the cell, or what it does is it goes and activate a protein, a cytosolic protein called GSK3. GSK3 is glycogen synthase kinase 3. Normally, this GSK3 is in an active form inside our cell. And the function of GSK3 is conversion of GS to phosphorylated form or phosphorylating GS2 to phosphorylated GS form. Once the GS is phosphorylated, it gets inactivated. GS means glycogen synthase. So normally what GSK3 does is it making GS as an inactive form or keeping GS in an inactive form. But once insulin comes and interact with the receptor, this GSK3 get phosphorylated by this PKP and the GSK3 get inactivated. Once GSK3 is get inactivated, now there is nobody to phosphorylate GS. Normally, GSK3 is phosphorylating GS and making it inactive. Once insulin comes and interact with the receptor, GSK3 itself is get inactivated. Now GS is ready for its function or GS is become active. What is the function of GS? The name itself implies that glycogen synthase. What it does is it convert glucose to glycogen. From where the glucose comes? Glucose comes from the bloodstream via LUT4 pathways. The another function of PKB is the opening of LUT4 channels. Actually, these LUT4 channels are present within the vesicles inside the cell. What PKB does is it causes the exocytosis of this LUT4 channels and the opening of LUT4 channels and causes the entering of glucose into the cell. And those glucose which comes into the cell are converted to glycogen by this glycogen synthase. I already told that there are two types of responses, right? Biochemical response and gene level response. This particularly indicate the biochemical response, right? Because here the glycogen synthase is kept in inactive form by cell itself or our body itself. Why? Because in normal time we don't need to convert glucose to glycogen because we need energy. We need energy. For that we have to do the glycolysis each and every cell in all time, right? So we need glucose as such. But when the glucose is in excess, on that time only we need to convert glucose to glycogen. So normally the enzyme which is responsible for glucose conversion or the formation of glucose to glycogen is keeping in an inactive form. When there is a need, on that time only it is converting to the active form. So this is the biochemical response of the insulin pathway. And previously we discussed about the gene level response where the cells that enzymes or proteins which are responsible for cell cycle is creating or making on that time only. So that is actually the gene level response of insulin pathway. So this is here we can see that the receptor is the first molecule which is making the process or switch on the process. So here the receptor itself showing an enzymatic activity. That's why these pathways called receptor enzyme type signaling pathways. Now the next one is where the receptor enzyme type where the receptor has no intrinsic enzymatic activity. Where the pathway is the same as that of this RTK pathways, but there the receptor is not having any enzymatic activity. The best example for such kind of pathway is JAK-STAT pathway. JAK means Janus kinase and STAT means signal transducer and activator of transcription. So in JAK-STAT pathway, we can see RTK receptor, same type of RTK receptor, but that RTK receptor is not having that enzymatic activity, where the receptor only acting as a docking site for the target molecule. This JAK-STAT pathway can be seen in EPO signaling or in stem cell signaling or in kidney cell functioning and all. We can see this kind of JAK-STAT pathway, where the receptor that mainly in the case of EPO signaling, EPO means erythropoietin signaling. Erythropoietin is a hormone which is produced from our kidney. So in EPO signaling, the EPO receptor, which is essentially an RTK receptor, this receptor normally existing as monomer. That is the hallmark of this RTK receptor. They are normally existing as monomers, except this insulin receptor that we already discussed, right? So this RTK receptor, this EPO receptor, which is existing as monomer, when there is EPO means erythropoietin, then only that receptor undergo the dimerization. Here the EPO mediated dimerization is actually coming under the class of ligand mediated dimerization of the receptor. Once EPO comes and bind with the EPO receptor, the EPO receptors undergo conformational change and they get dimerized. During the dimerization process, they create some docking site within the receptor. This docking site actually mainly for binding with the Janus kinase proteins. Janus kinase means a cytosolic protein. Normally, they can't interact with the EPO receptor, but once it gets dimerized, this receptor creating some docking site for Janus kinase, so that now Janus kinase bind with the receptor. In this case, the receptor has no enzymatic activity. So, it is acting only as a docking site. Okay. Now, once JAK bind with this receptor, just JAK phosphorylating the receptor. In the previous case, the receptor itself undergoing a trans-autophosphorylation. But in this case, the receptor lacks that type of activities, so that here the Janus kinase, which is a kinase, this kinase causing the phosphorylation of the receptor, and this kinase phosphorylating the receptor, and to that phosphorylated receptor, now another cytosolic protein called STAT comes and binds. The STAT once interact with the receptor, it has got a phosphate group from the receptor. Actually, the receptor is not responsible for phosphorylating the STAT. The JAK, which is attached with the receptor, is responsible for phosphorylating the STAT. Now, in this pathway, STAT doing two functions. First, it is phosphorylating the receptor. Second, it is phosphorylating STAT. Once STAT get phosphorylated, the STAT undergoes a phosphorylation reaction. Actually, why it is undergo conformational change once it get phosphorylated means it is always because of the reason that it has got a negative charge. This negative charge creating some conformational change within the protein, that we know that protein has got lots of conformational changes like quaternary structure, tertiary structure, and during that structural formation, when an additional charge is added to the protein, it's all conformation get modified. That is actually happening to this molecule. Okay. Anyhow, once this STAT is phosphorylated, the STAT undergo a conformational change. Because of that conformational change, an additional docking site is forming inside the STAT. When we analyze the structure of STAT, we can see some coiled coil structure within the STAT molecule. This coiled coil structure is actually responsible for the formation of this docking site. Okay. Once STAT forms a docking site, such kind of STAT monomers get dimerized and form a STAT dimer. This STAT dimer now can cross the plasma membrane. Till now they are in the cytoplasm. They can't cross the plasma membrane. Why they can't cross the plasma membrane means there is no conformational change in that STAT molecule. Once they get dimerized, they has got a negative charge, extra negative charge, so that they can now cross the plasma membrane. Once they cross the plasma membrane, they reaches the nucleus, where it bind with the EPO specific genes. EPO is erythropoietin, and which is responsible for this stem cell proliferation. So those genes which are responsible for proliferations are get activated by the STAT monomer. Actually, the STAT dimer acting as a transcriptional activator of EPO responsible gene and causes the transcriptional activation and resulting in the transcription and translation of genes involved in the STAT, that EPO formation or the cell proliferation. So in this case, there is no enzymatic activity for the receptor. In the previous case, there is an enzymatic activity. Here there is no enzymatic activity. These are the two major pathways involved in the biosignal. The rest the three pathways are there. The next one is GPCR, G protein coupled receptor signaling, where the receptor is actually coupled with the G protein. I already told you that G protein is nothing but a protein which is binded with or which is attached or coupled with the G protein or guanosinucleotide protein. A receptor which is associated with the G protein is called a G protein coupled receptor. In this kind of pathways or in GPO pathways, the essential point is nothing but once the ligand bind with the receptor undergoes a conformational change. This conformational change ultimately changing the conformation of a G protein. Normally in inactive form, G proteins are associated with the GDP. That is the inactive conformation. Once G protein get activated, their GDP is replaced by GTP. Be very careful, that is GDP replacement. There is no phosphorylation reaction. Okay. Now, once its GDP is replaced by GTP, that GTP form of G protein become activated, and now the GTP protein causes the activation of a series of preceding molecule. This kind of reaction is we can see in the case of G protein coupled reaction. The next type of signaling is integrin signaling. This kind of signaling we can see in proliferating cells like stem cells, or in case of embryonic cells, we can see this kind of cells reactions, integrin signaling. Actually, this integrin signaling is responsible for cell-cell addition, that is one cell communicating with other during the addition reaction. This kind of signaling we can see, where we can see a two-way signaling. Normally, I told you that that signaling is of two types, that is in-out signaling or out-in signaling. But in the case of integrin signaling, you can see an in-out signaling. Both in-out way of signaling you can see in the case of integral signaling. Last one is actually nuclear receptor signaling, where the receptor is actually present within the nucleus. In the case of integrin signaling, or in the case of RTK, or in the case of receptor without enzymatic activity, all these cases, the receptor is actually transmembrane in nature, or structurally they are transmembrane. But here, the receptor is present within the nucleus. Within the nucleus, either maybe in the nucleus or maybe in the cytoplasm, that doesn't matter, but its action is always on the nucleus. Essentially, this receptor is present within the cytoplasm. Since it is present in the cytoplasm, it can interact with only that kind of ligand which can cross the plasma membrane, or it is meant only for ligand which is hydrophobic in nature or lipid lovable ligand. Those ligand which coming under this class is steroid ligands. Steroid ligands are normally ligand which can cross the plasma membrane. These ligands, steroid ligands means mainly ligands for estrogen or progesterone, such kind of, or androgen, such kind of hormones. Ligands are mainly hydrophobic ligand. They can cross the plasma membrane and they can interact with the receptor present in the nucleus. And these ligand always causes that actions on nucleus, or their function is always associated with the gene level response, or the transcription and translation of the protein present in the in our biological system. So these are all about the classification and the detail about the biosignaling process that is taking place in our body.